Good evening, everyone. Welcome to this evening's Broad Discovery Center talk. My name is Tom Ulrich. I'm the Associate Director for Science Communications at Broad, and it's my pleasure to have you all here with us for a little while tonight. For those of you who might be joining us for the first time, the Broad Discovery Series has a bunch of different goals that go along with it. We explore, I'm usually better at this. You'd think I do it 4 times a year. We explore the genetic and biological roots of health and disease. We explore also the mechanisms that govern how our cells and bodies work. We talk about new technologies that are changing what's possible in science. And we also are talking about the progress that's being made to translate new discoveries into treatments for both common and rare diseases. It's my pleasure tonight to introduce Niall Lennon. Niall is the Senior Director for Broad's Genomics Platform. He's also the Associate Director for Broad's Gerstner Center for Cancer Diagnostics. And in addition, he's also the Chief Scientific Officer of Broad Clinical Labs, which is a wholly owned subsidiary of the Broad Institute. Niall's going to talk tonight about the opportunities that the constantly increasing speed and dropping cost of genome sequencing are allowing for healthcare both now and in the future. If you have any questions for Niall tonight, I encourage you to flag one of our mic runners during the Q&A portion of the evening, and they'll bring a microphone right to you. If you're watching online, I'll be keeping an eye on YouTube chat, and so if you just enter your questions there, I'll make sure to do my best to try to work you into the conversation. If you're a scientist or a Brody with a deep technical question for Niall, I'm going to encourage you to please approach him during the reception after the Q&A so that members of our local community have an opportunity to have their questions answered. And lastly, speaking of receptions, I invite everybody here in the room to join us after tonight's talk and for a reception in the Broad Discovery Center, right back out the doors here, where I encourage you to explore the center's exhibits and interactive elements. Come on up, Niall.
All right, good evening. Can you all hear me? All right, yes, the microphone's working. Fantastic, great. Okay, so I am delighted to be here this evening to talk to you about what the title says, which is essentially how the technologies that we're using at the Broad, the data we're generating here, and the biology that we're increasingly understanding might play into a future healthcare system that incorporates genomic medicine. So in my title, I used a word that should make us feel all warm and fuzzy. But actually, I imagine for many people, it's actually a cause of stress. Healthcare, right? Who wakes up at 3 in the morning and you're wide awake and your mind is rushing with all of the topics of the day? I do sometimes. And sometimes those thoughts are like, why can't I play golf better? Sometimes those thoughts are, what if I get sick? What if one of the kids gets sick? Today, it feels like the healthcare system has become a system that we never want to have to interact with. And that's probably not where we want to be. But we shouldn't despair. I do want to point out there's some things to give us hope. And some things that, when we think about what's possible in modern healthcare, that we should be really excited about. Just some examples at a high level. We do now have the capability to see disease at the genomic level. I'll describe more what that means. But more importantly, we're actually increasingly able to actually target with therapies the exact molecular causes of disease. Whereas historically, we may have treated the symptoms of disease, how diseases show up. Through the technologies that I'll describe and the insights, we are increasingly being able to see what's causing that disease at the molecular level and designing therapeutics that go after that, and that is more effective. And that now also for the first time through technologies developed here and elsewhere, we are able to not just identify those underlying changes in our genomes that might be causing disease, but for the first time we can actually start to think about editing those genomes to fix that underlying error. So these are all exciting developments, and they've happened just over the last 20 years. In fact, they've started to happen just when I joined the Broad Institute 20 years ago. Coincidence? Yes, definitely a coincidence. However, there are some things that I think are not coincidences. I think there's some examples that we should look at where there is a clear cause and effect thing happening, right? And I want to point us to that time when we collectively decide to invest in audacious projects, in really difficult things, to generate, to achieve some really hard to achieve goal, and that the effect of doing that has been positive in our history. So one example being the Apollo program, right? To send astronauts to space, to land on the moon and come back. That program ran for about 13 years, from 1960 to 1973. The government invested about $25 billion in that program, all told in that time frame. And estimates now suggest that for every dollar we invested in that program, we got a return on that investment of about $7. And that return on investment came from solving the problems that we needed to solve to achieve the goal. And it resulted in advancements in material sciences, in batteries, in portable tools, in memory foam. Hey, I've got a memory foam mattress, cool. I didn't realize it came from the shuttle program. But the point is, when we do something like this, we invest public funds in something really difficult, we actually receive returns on that investment. So another large-scale investment that we've made as a country was the Human Genome Project. It's not something that probably most of the general public holds in their mind the same way that sort of the Apollo missions are known, but it similarly was actually technologically extremely difficult when it was started, when it was contemplated first. Sequencing the 3 billion letters of code that make up our genome at the time this program was started was extraordinarily difficult task. The program took about 13 years as well to complete. It ended up being about $3 billion of public investment, and the National Human Genome Research Institute estimates that we've seen about a return on investment of about $5 for every dollar invested in that program. So again, an audacious idea that we should sequence the human genome has resulted in a proliferation of fields that now use those technologies and the information that we can now generate, not just in biomedicine, but also in forensics, in genealogy, in agriculture. So there's a ton of spaces where now we use the technologies that had to be invented to solve the Human Genome Project now in everyday life. So I'm going to not use too much jargon tonight, but I've already mentioned the word genome a few times. So just to level set, the genome is basically the genetic material of an organism. Predominantly, I'll talk about human genomes, but actually in our community, there are folks who study human genomes. We study microbial genomes, viral genomes. There are industries that look at plant genomes, animal genomes, but largely they're all using the same sets of technologies to do that. And so the other term is variant. And so in the past, we might have used terms like mutation, but today we would say a variant is essentially any difference that's in our genome between two different individuals. And part of the reason to actually do the Human Genome Project was there was an understanding that variants, that differences in our DNA, in our genomes, might be underlying not just sort of our traits, how we appear, but also might be underpinning some diseases. But until we actually sequenced the genome, there was no way to actually know which variants in our genomes are totally harmless and which are the ones that actually could be the triggers for disease. So that was the reason to start doing genome sequencing. And the other important point is that to understand variants across many people, of course, you can't just do the human genome once. You actually have to do it many, many times. Ideally, we would have everyone's genome sequence, and then we would know all of the variation in the world, and we would be able to catalog what variants are actually disease-causing or disease risk increasing, and what ones are totally harmless. And so the other thing, I guess, to think about is, unlike space travel, which is still pretty expensive, it is now primarily, the purview of billionaires and minor celebrities. Sequencing human genomes has actually continued to go down in cost. So from that first human genome, which again, overall cost about $3 billion to sequence, to understand the code of, through technological advances over the last 20 years, we're now at today, if you come to the Broad Institute as a researcher, you can get a human genome sequenced for a few $100s. And unlike that first human genome, which took 13 years to sequence, today, routinely, we can do it actually in days. And I'm going to tell you something special that happened about a month ago that might indicate that in the future we'll be able to do this routinely in hours. So today at the Broad, we actually sequence thousands of human genomes every week. In fact, in our group, at the genomics group at the Broad Institute, we've now sequenced over 870,000 human genomes. And if you think about the idea, again, of sequencing the genome, there's also a lot of information in the genome that we don't yet know fully what to do with. The genes that make up all of the proteins that sort of make up our body are actually encoded in about 2% of the information of our genome. And if you just sequence that portion of the genome, it's known as exome sequencing, the expressed part of the genome. And of exomes, we've done over 1 million of those. So the technology and the drop-in cost has allowed us to now expand the scope of how many genomes or portions of genomes we can routinely sequence. And why do that, I guess, right? So I hinted at, before that you can't just sequence one human genome and start to really unlock all of the insights of biology that are locked in there. You actually have to sequence lots of genomes. And so the genomes, the over 870,000 genomes that we've sequenced, and hundreds of thousands that have been sequenced in other parts of the world, have been feeding into research programs funded by federal government, by philanthropy, by disease foundations, by pharmaceutical companies and biotech, to really push forward all of the possible applications of human genome sequencing. And these are some of the application areas I want to sort of describe to you in more detail tonight. And these are the areas that actually, where people are showing the utility of genome sequencing across their entire lifespan, from birth all the way through aging. And so I'm going to dig into a couple of examples. By no means is this an exhaustive catalog of all of the activity that's happening, but I just want to give you, in certain parts of this sort of lifespan space, where we're at and where we might be going. So think about newborns. So babies just born. Most states, if not all states in the US, have state-mandated newborn screening programs. So we test babies. We do a little heel prick, get a little bit of blood, and we test babies for some small number of diseases that are important to know about as soon as you can possibly know about them, because you want to take an action that would avoid that baby getting sick in early childhood. So these are known as newborn screening programs, and they're typically managed by departments of public health. So several studies now, over the last few years, have been investigating, rather than this sort of small suite of 30-ish largely biochemical types of tests that we do on those newborn heel pricks, should we be doing whole genome sequencing? Two notable studies. One is the Guardian study run out of New York City, where they sequenced many thousand newborns. And initial results they published a year or so ago showed that by doing whole genome sequencing, you would find many, many more conditions that geneticists would agree you would want to know about in a newborn than you do by traditional screening methods. So this idea of like establishing sort of clinical utility. Another study, and good news for us in the Boston area, that program, that Guardian study, was run by an investigator out of Columbia University, Wendy Chung. Wendy Chung is now the chief of pediatrics at Boston Children's Hospital. So she's been bringing this concept to Massachusetts as well. Another study closer to home, run by Dr. Robert Green out of Harvard and Brigham and Women's Hospital, was called BabySeq. And the sequencing of these newborns actually happened in our clinical lab here at the Broad Institute. And that found, again, in a smaller cohort of several hundred newborn families, that they found disease risks in about 11% of the babies where we sequenced them. And that, importantly in that study, they were also addressing sort of the family response to the information. Because one of the fears of doing this type of testing on a newborn, certainly a healthy newborn, is like, will it cause anxiety to the family? Will knowing this information cause families to be super stressed out? And they did surveys of their families and found that that wasn't necessarily the case, that typically families were happy to participate, and the ones who found out actionable information were very happy to have it. So that was great. So these have sort of set the stage. Going forward, there's actually a new program funded by NIH called the Beacons program that is going to try to understand the feasibility of offering this type of whole genome sequencing as a first-line test in newborns on a multi-state level. So the sort of technical components of doing this and the initial impact studies have been done. The real challenge now is actually the implementation science. It's like, how do we actually roll this out into states where this is managed by departments of public health who are not typically managing high complexity molecular testing programs. And so this program will actually seek to bring this out into more states. So this will help us in the future, and if the results are positive, we might see a national program at some point in the future. Another case in the sort of the newborn field is that unfortunately some small number of babies are born who actually have a serious problem, and they end up in the neonatal ICU or the NICU, and these are very sick babies, can be very sick babies, and there again, you're not dealing with screening for disease in a healthy newborn. Now you're looking for, can we get to a diagnosis for a very sick baby? And obviously you want to do that as quickly as you can. And so a study, again, a landmark study, this time run out of Rady's Children's Hospital in San Diego, run by Dr. Stephen Kingsmore over there, again, looked at the feasibility of running whole genome sequencing in this NICU cohort and understanding what is the benefit. And actually he found that there was a huge benefit, and that the information you gain actually can decrease the length of time it takes to get to a diagnosis. Importantly, they also looked at what would be the healthcare economics of doing this, and saw that you might actually reduce the cost, the healthcare cost, by doing this testing up front. Those savings are driven by fewer hospital stays or days in the NICU, because if you get to an answer sooner, you can get out of the NICU sooner. And those are very expensive stays. Fewer major surgeries. You can avoid a major surgery if you can get to a definitive diagnosis sooner and not do unnecessary procedures. So all of that together indicates that it might be really valuable to do whole genome sequencing in the NICU setting. Based on this and other studies like it, now clinical commercial laboratories have started offering what they call rapid whole genome sequencing programs, clinical whole genome sequencing programs that can be used by ICUs and NICUs around the country. With current technologies, they're still taking between 2 and 5 days to actually get you that answer back, though, which is really fast. Again, 2 and 5 days, really fast. But in the NICU, every hour matters. And so we were fortunate a few months ago to actually engage in a study to assess, could we do it even faster? So this was a collaboration between us, Roche, which is a large pharmaceutical company, but also has a diagnostics arm, Roche Diagnostics, which is a very large entity in its own right. And within that arm has a small group that's been developing a novel DNA sequencing technology. So we worked with them using this novel technology and our friends at the NICU and Boston Children's Hospital to run a small study where we showed, and the plot sort of shows think about this as sort of the a clock of a day, where we were, so Monica Wojcik, who is the neonatologist at Boston Children's, enrolled families into this as a research study. So it's not a clinical diagnostic test, it's a research study. Their baby samples were already being sent for standard of care clinical testing. But as a research arm, we got additional blood from the NICU babies, the sick babies, had that collected by a courier at Boston Children's Hospital. We rushed it out to our clinical lab, which is actually located in Burlington. And then we started working on that blood sample using this new technology. By about noon on that day, we had from the blood sample extracted genomic material, run it through this novel sequencing technology, and gotten to sort of the data files, the computational files that contain the variants within that baby's genome. And then we started what's called tertiary analysis. That's where trained clinical geneticists look at all of those variants in special software that allows them to sort between those variants that we know are most likely harmless and those variants that have been associated with disease. And they have the phenotype, that is, what are the symptoms that the baby is exhibiting. And by looking at the symptoms and the variants in the genome, they can sort of say, is there something here that actually might be causing these symptoms in this baby? So in 2 of the 7 cases, we actually located what we think is likely the pathogenic variant, the variant that it was causing those babies' symptoms. And importantly, if you look at the clock, what happened was we were collecting blood at 6:30 in the morning, and in the earliest cases, getting the neonatologist the answer by around 2 p.m. that same day. It had never been done before. And so we published this in New England Journal of Medicine about a month ago as a feasibility to say it might be now possible in the future, once this technology is commercially available. It's still not commercially available, but once it is, as soon as next year, we may be able to now routinely deliver whole genome sequencing the same day. And that would really radically change the thinking of neonatologists in how they create a care plan for the sickest of babies. And just for fun along the way, we actually picked up a Guinness World Record for the fastest human genomes ever sequenced. So if you remember, we've come a long way from 13 years. In this study, we sequenced a genome in less than 4 hours. So from that DNA to those variants in less than 4 hours. So the technological advancements have been, mind-blowing over the last decades. All right, so the next place in our lifespan where diseases can show up is in childhood. And a lot of childhood diseases are rare diseases. And so we call them rare diseases because each sort of disease that shows up doesn't show up in a lot of children, but in aggregate, there are a lot of rare diseases. In fact, there are over 8,000 rare genetic diseases now described. So the burden on the population is actually quite large. And fewer than 500 of those rare genetic diseases have any treatment available. So understanding the causes of rare diseases is very important. And again, rare diseases are often have genetic causes. And so we've been looking at, again, with collaborators at Boston Children's, and there's many programs at the Broad that look at rare disease. How can we bring these new technologies to look at the underlying genetic causes of disease? Sometimes the sort of standard genome testing we do is not enough, and we need to go to additional data types, different types of doing genome sequencing to find that sort of missing cause. Because what you're really trying to do in the rare disease case is decrease what's known as the diagnostic odyssey, which is that families who have a child with rare disease often go through years of like incremental testing one gene, testing the next gene, and it could take years to get to an actual definitive diagnosis. If you can do that sooner, again, you have many benefits for the family. Just having an answer is really important. It helps them with planning. Is it likely to happen in subsequent children? It helps them find support groups and other people who may have the same diseases. And then also, start to figure out, are there clinical trials we can enroll in? So it's really important to get to that diagnosis as soon as possible. And that's been a lot of focus of whole genome sequencing methods. Other programs that we've been running with disease foundations include the Simons Foundation, which runs a large autism research program. There are actually over 300,000 now participants, both participants with autism and their families. And we've been doing whole genome sequencing on many of those individuals to, again, understand some of the underlying genetic causes of autism. A future-looking program that started at the Broad Institute under Anna Greca, a researcher at the Institute, is called Ladders to Cures. And this is a really tantalizing prospect that Anna has described in detail in other talks, but where, if you think about these 8,000 rare genetic diseases, it's entirely possible under sort of growing evidence that you might not be actually working with 8,000 completely different diseases that have just sprung up independently, but that if you look deep enough, that they actually might sort of whittle down into smaller numbers of sort of systemic kind of network effects where, something that's happening in a node, in sort of a, underlying system in the body is manifesting in lots of different ways and getting diagnosed as different and showing up as what we call different diseases. But actually the fundamental molecular thing, this pathway, is actually common across many of those diseases. So if you can if you can discover and describe those pathways, those nodes, you might be able to come up with more effective therapies for more diseases at once, rather than trying to tackle all of them individually. And this uses other aspects of technology that we have. really looking at the single cell level, looking at individual cells and many, many cells, and engineering them to display some of the variants that are showing up in these diseases, and seeing what happens across different variants. Do the cells start to do the same thing, different things? And so this is really an exciting new way to get to address rare disease research. Of course, chronic diseases and common diseases are the largest cause of sort of mortality and morbidity in the world. And there are many programs as well, of course, that address these diseases. There are programs funded by the NIH, which address lots of common diseases using both genome sequencing or genomics, as well as many other omics. When we talk about genomics, there's also really looking at the protein level, proteomics, looking at the sort of chemical signatures that are on top of our genome, or epigenomics. There's metabolomics, there's transcriptomics, there's lots of omics. And so we're increasingly understanding that for cohorts of people with disease, it's really more powerful to generate as much omic data as we can, because that gives you a much fuller picture of what's happening, again, at the molecular level. Another really important area in common disease research at the Broad is coming through a philanthropically founded center at the Broad, the Stanley Center for Psychiatric Diseases. That group has been doing large-scale population sequencing across the world of different populations that have psychiatric diseases, schizophrenia, bipolar disorder, major depressive disorders. And they've actually, through this large-scale genomic data generation activity, started to uncover biomarkers or those little signals in the genome that strongly can be linked to these devastating mental health issues. And in some cases, these are the first sort of genetic targets that we've identified in decades in these diseases. And so this gives the idea that there's hope to start to generate better, more targeted therapies for major psychiatric diseases. Again, looking forward, one of the newest centers created at the Broad is the Biology of Adversity Project. This is a really interesting area driven by Jason Buenrestro at the Broad Institute, which is to understand how adversity can actually lead to chronic health problems. And so, we've all been taught that the DNA is your blueprint, it's in every cell of your body, and it's sort of, we kind of imagine it's sort of fixed. Once you've got your DNA, you've got your DNA. But actually, it turns out that things that happen to you in your life can actually leave a mark on your DNA. And this is part of that epigenomics, the idea of molecular scars or little signals, little chemical modifications that can get imprinted on your DNA and propagate through your life. And we're increasingly understanding that these molecular scars, if can lead to later life development of chronic diseases, heart disease, diabetes, major depressive disorders, post-traumatic stress syndrome. And so Jason's group and the Biology of Adversity Project is seeking to really do what the Stanley Center has been doing in the psychiatric disease research area, but looking at these molecular scars in cohorts who have had adversity. So these are childhood traumas, understanding cohorts of people who have had some traumatic event, and seeing, can we find these markers, and are they actually things leading to chronic disease? So this is a very fascinating area that could be really powerful to understand a lot of chronic disease. All right, cancer, of course, is one of the sort of prime examples of where we've seen a major benefit of being able to do molecular genomic profiling. Increasingly today, patients diagnosed with cancers will have their tumors genomically characterized. And so when we think about your genome, the genome of your body, sort of the quote-unquote normal, healthy genome, in cancer, for the most part, that genome has somehow become mutated, transformed in some way that has led to this uncontrolled proliferation, and that shows up as tumors. And so there are many programs at the Broad and elsewhere to really catalog the variants that are associated with different cancer types. And these are, one way you can do that is to take a blood sample and a tumor sample from an individual with cancer and sequence the genomes or portions of those genomes from both of those. And what you'll find is that in the sort of the quote-unquote healthy non-tumor cells, you'll have the person's underlying genome, but in their tumor cells, you'll find this abnormal genome. And by cataloging all of the ways that it has become abnormal, you can uncover these molecular pathways that are leading to cancer. And this is the underpinning of most targeted molecular therapies now, precision medicine therapies for cancers that have been so transformative in changing the prognosis for most cancers. And so there are programs across the board cataloging these variants in different tumor types. Also, again, using this idea of single cell technology to create sort of tumors or engineer in modifications that represent a cancer-like mutation, and then be able to quickly screen what do different small molecules, what do different medicines, what do different drugs do to those cells. Can we find even better targeted therapies? And then also the idea of overcoming resistance. Sometimes people get a therapy for their cancer goes away, but they develop a resistance and the cancer comes back. So understanding those programs of resistance is also really important. Another center at the Broad, the Gerstner Center that I've had some involvement with, is doing similar things, but actually in the sort of the burgeoning field that's come in the last couple of years of understanding that actually a tumor in your body, because of the nature of cancer, tumor cells are often dying. And when they die, they release their genetic material that essentially gets drained into your blood system, which is how your body essentially cleans out. And so that by looking at blood samples, just taking a blood sample, you might be able to find what's known as circulating tumor DNA. That's little pieces of DNA from the tumor that are actually circulating in the blood. And so this field of what's known as sort of liquid biopsy. So a traditional tumor biopsy would be there's a tumor, let's take a piece of that tumor out, that tissue, and look at it. The field of liquid biopsy is actually taking a biopsy not from the tumor, but from the blood or other liquids. And this is particularly important where you've had a patient who's had cancer, their tumor has been removed. You've taken away the cancer. So now there is no tumor to biopsy anymore. But what you really want to understand is did we get all of it? Or is there another tumor that we haven't seen yet? Is there metastasis? These are really important things because these are the things that actually lead to a lot of mortality in cancer patients. And so being able to detect, can we find any signals of lingering cancer? This is a field known as minimal residual disease. Is there any small signals of tumors circulating in the body? And so now, by taking blood samples at some period after the person has had surgery or treatment with curative intent, there's a whole field of, can we detect any cancer? And if you don't, that's great. These methods are getting now more and more sensitive, thanks to the work of folks at the Gerstner Center and elsewhere, to see the earliest possible time, if you do detect it again, that's the time to take an action. Because detecting it and having it see that it's actually coming back is the first signal that something is going to come back. And so the earlier you can have that minimal residual disease detected, the more powerful intervention will be. So that's a huge area now in the field of cancer research. Looking forward again, just in the last few months, locally again, we've been able to bring together the best of our cancer research community and our local hospital community with one initiative called BrightSeq, which is again a collaboration between Dana-Farber Cancer Institute, Boston Children's Hospital, and the Broad Clinical Labs here to develop a suite of pediatric cancer assays. So there are many now, both companies and labs that do adult cancer testing, because that is the vast majority of cancers that show up in the world. The pediatric cancer is thankfully quite rare, but what that means is from a commercial perspective for these companies, it's not actually a super viable business. And so you have less development of assays really specifically developed to detect or monitor pediatric cancers. And so we've been, as part of this collaboration, we will be developing both solid tumor and liquid biopsy-based assays for pediatric cancer diagnoses and research. All right, moving on. So a biobank. Has anyone been asked to be a participant in a biobank, as far as you know? So if you've got the Mass General, you might have at some point been asked, might have seen.
Is it like all of us?
Yes, exactly, right. So a biobank is essentially a program that creates a secure, consented repository of biospecimens and people's information, right? And so the way to really drive forward many of the things that we've just talked about, right? Understanding that catalog of human variation, what ones cause disease, what ones don't cause disease, is to actually have information from lots and lots of people. What we're realizing, of course, is that, again, fundamentally, at a very simplistic level, when you sequence a genome, you're trying to understand the variants that you find in that genome. And there's millions of them. In all of our genomes, a variant, something that's different than somebody else's genome, there's millions of them. So if you're just looking at a computer file with 1 million or 3 million variants in it, and you're trying to decide which one of these 3 million things might be causing this person's disease, that's really hard, right? So you need to have knowledge bases, computer systems, software that can sort through all of that and annotate it and say, in all of the people we've ever seen, this variant, this difference in a piece of DNA has never been known to cause a disease. That would be ideal if we could do that for all 3 million. But of course, to do that, we actually need to see all of the variants that exist in the human population. And it turns out, biobanks are a great way to start cataloging that. Because biobanks try to enroll people, some people who have disease, but ideally just regular people who, we've all got something going on, but nominally healthy people, right? If we can all contribute our information, both our genetic information, surveys about our health, our medical records, information about us that goes into a large biobank-like knowledge base, we can really start to understand, the impact of that variation. A biobank that got started early and has been very successful is the UK Biobank. The NHS and the UK government have been very forward-looking and investing in biobanks and genomics. And so they've got a really mature biobank with over 500,000 individuals, with this sort of linked information about health records and activity and everything else. And so that's been very powerful to understand some of the variation in humans. But what we're realizing, of course, is that variants in our genome don't just account for sort of our hair color and our eye color and our, whether or not we have asparagus pea. They also account for, they actually also come from our genetic ancestry. So different human populations will have different variants. And so some of those differences between us come from our genetic ancestry. And it turns out that if you look at a variant in one population and say, ooh, in this population, we see this variant, and then that person also has this disease. So maybe that variant causes that disease. But if you look at enough people, maybe you find in a totally different human population, that variant exists and is quite common, and none of those people have that disease. Well, now you've realized, actually, that variant can't be the one causing the disease. So you actually need to catalog people from all types of genetic ancestries to really understand what variants are important to look at. And so if you look in UK Biobank, and you look at a variant and you want to predict something about my genome, that would be fairly accurate. Now, I'm from Ireland. Ireland is not part of the UK. But genetically, we might not want to admit it, but we're sort of similar. So it's going to be accurate for me because most of the UK Biobank is from Northern European white individuals who live in sort of that UK area. But it's not going to be as accurate for somebody who comes from Africa or somebody who comes from Asia. So we actually have to start creating biobanks that reflect everybody. And so All of Us is one of those programs that in the US is trying to catalog at least 1 million US residents and capture sort of the genetic diversity and socioeconomic diversity of this country. Because unlike many countries in the world, US traditionally has been this sort of what we would call like the boiling pot. You have immigrants like me who come over from Europe, and you've got other people who come from all over the world. And then they meet somebody and they have children, and so it turns out your grandfather's from Ireland and your grandmother's from the West Indies. So your genetic ancestry now is actually the combination of two entirely different human populations' genetic ancestries. And so from a geneticist perspective, we would say you're admixed, right? Your genetics are mixed across different genetic ancestries. And so we need to capture we need to capture all of that. So it's not just useful to sequence a bunch of people from Europe and a bunch of people from Africa and a bunch of people from Asia. We also have to sequence just all the people who've all kind of, had offspring together. So you really need to capture, it's a numbers game. You need to capture as many people as you possibly can to build up the knowledge base in a biobank. And then you need to associate that with a lot of interesting information about their health and their activities and all of that to really build up this picture of what's going on. And so that was the program, the All of Us program. It's enrolled over 800,000 people. It was started many years ago with a goal of enrolling 1 million and sequencing all of their genomes. It's enrolled about 800,000. We have sequenced over 500,000 of those participants' genomes. Many of them were done here at the Broad Institute. The program has run into some federal funding issues. And so the future of that program is uncertain. And I think that would be a tragedy. More locally, actually, if you attend a hospital around here in the Mass General Brigham network, Mass General Brigham has a pretty actually active and pretty good biobank program as well. And that's enrolled over 160,000 people from the Mass General Brigham Hospital network. And that's also a really powerful data set to start to look at, again, associations between your health and your genome. So as well as being a principal investigator on the All of Us program, because we generate a lot of the data, I believe in this stuff. I'm a participant in the program. My genome was sequenced as part of this program. My wife's genome was also sequenced as part of this program. Early in the program, when it had more money, it was also returning some health-related information to participants. And so you would get some information back about your health risks. There are some things that we can tell that are important to know. My wife actually found out that she has what's known as Lynch syndrome from participating in the All of Us program. And that means that she's at an increased risk, potentially, of colorectal or ovarian cancer. And so now she's brought that information to her PCP, and now she gets more frequent screening for those particular conditions. So that idea of just learning something about your genome that you didn't know can completely change your healthcare. So to kind of recap from a biobank perspective, what can you do? You can do lots of things with it. A rich biobank that has lots of sort of genomic and other omics and associated health information can help us catalog this human variation. It can help us now, because if you actually enroll hundreds of thousands of people, you're going to have lots and lots of people with any one disease. Certainly common diseases are going to be common. So you're going to have lots of people with cardiovascular disease, lots of people with cancer. And so you can then look in this program and say, well, what's the same about all of those people with that disease that's not in the people who don't have that disease, and start to do those genetic associations. The other tantalizing thing that I think, looking forward, is that by having all of this data, we can start to get really fancy with the sort of the math, right? And we can start to create what's known as risk models, right? And so this is the last sort of area that I'll cover, which is preventative medicine. The idea that treating disease, we've gotten really good at treating disease, and we've got world-class hospitals here in Boston. But it's a lot better to avoid getting the disease in the first place if you can. And that's actually fundamentally, longer term, going to be better for the whole healthcare system if we can avoid treating the symptom, but actually, avoid getting the underlying disease at the beginning. And a Millikan Institute analysis estimated that if we could just prevent or better manage just 7 common diseases: cancer, diabetes, hypertension, stroke, pulmonary conditions, and mental illness, we could reduce the US treatment costs by about $200 billion per year. And we'd save about $1 trillion per year in lost productivity. So prevention is clearly better than cure, but of course it's difficult. And there's lots of things that go into preventing disease. And it's hard to measure because you're looking for the absence of something if you've prevented a disease. But this is something that I really feel passionate about, and I think has driven me and my own sort of journey into this kind of science. When I was 16, my father had a heart attack. He was 46 at the time. I was 16, so I thought, wow, he's really old. Then I turned 46 3 years ago, and I had 3 children under 12, and I went, I don't want to have a heart attack. I'm really young still. My father survived, and he's actually alive and kicking. Has had several stents and bypass surgeries since then, right? So clearly, I was really focused on the idea of what is my risk of cardiovascular disease? I'm really cognizant of that because I've got this family history. So I'm like, I really want to know about that. And so when you think about this idea of what goes into this mental model of what is my risk of developing this disease, of having a heart attack? And so there's lots of things. We've touched on this. One is your genetics, your family history, any other underlying sort of medical conditions you might have. There's these ideas. So the terms up here, polygenic and monogenic. without getting into too much detail, mono, as the name implies, is essentially there are some variants in some genes that are well known to be associated with disease. So we all might have heard about this idea of the BRCA1, BRCA2 mutations in breast cancer, right? This Angelina Jolie mutation. That's well known. That's known as a monogenic risk. If you have that one single variant in that gene, we know that's really strongly associated with developing that condition. And there's monogenic risks for lots of diseases. Polygenic risk really means this idea that there are not these sort of single sort of smoking gun, this one variant is really bad, but actually distributed throughout our entire genome. There are small, there are variants that have small or varying degrees of effect on our health, and that any one of them wouldn't really cause a disease. But in aggregate, if you had all of the wrong kinds of those variants together, you might actually be increasing your risk of a disease. And that's known as polygenic risk, because it's in lots of genes, okay? So the idea of, to get a full picture of our risk of disease, you need to understand monogenic and polygenic and your family history, and also social determinants. What is your access to health? What is your exposures? Are you living in an area with polluted air? That also contributes to our risk. So you need to get a big picture. So biobanks are a great place to start to aggregate all of this information and then to build these models. And these models can be super powerful, and basically they're algorithms that take all of these pieces of information together and come up with a risk and say, you are at an increased risk of this disease. You should take action. We were fortunate to be part of a study funded by the National Human Genome Research Institute known as eMERGE, or Genomics in the Medical Record, which actually sought to look at this idea of what happens if you report back to individuals their monogenic and their polygenic and their family history risk of some chronic diseases. We chose models that were mature for the diseases listed here. And across 24,000 participants and 10 clinical sites across the entire country, including both adults and pediatric cohorts, we sequenced, we calculated their risks of these diseases through these models, and then returned that to them through their healthcare providers. And again, the science is sort of there, but a big part of this study is actually the implementation science part of it. Like, what do they do with that information? What do they do? How does, not just the patient, what does the doctor do with that information? So this idea of polygenic risk for cardiovascular disease has been developed, and some of the seminal work on that came out of the Broad Institute several years ago. I'm a patient, and Broad Institute and researchers at Mass General that work with us published some seminal papers on this many years ago. And of course, I'm like, oh, I'm interested in cardiovascular risk. I'm going to go talk to my PCP, who is at Mass General, and say, what do you think about this polygenic risk? Should I get tested for polygenic risk for cardiovascular disease? He went, what's that? Right? He didn't have a clue. Because we're not training physicians who are in the field with 50-minute appointments on what polygenic risk is. So part of this study was actually, what about creating educational materials for the healthcare provider and action plans, and how do we train the healthcare system to actually be able to handle this information as well, not just the participants. And so this is, again, I think the next generation of what we're going to start doing is how do we start to distribute the more cutting edge models of risk so that people can start to take actions earlier. And so we've been fortunate, building on this study and other work that's been happening, to now work with lots of groups to try to get this information out there. So Mass General Brigham, again, across the river, Pradeep Natarajan, who runs the Preventative Cardiology Clinic at Mass General and is a member at the Broad, he's been really interested in this topic as well for his clinic. Because you can do all the risk factors of lipids and weight, exercise, smoking, but he wants to also capture the full risk, which means he wants to understand that polygenic and monogenic risk as well. So together, we worked on launching a clinical test now that any patient, and not just at Mass General, but any patient around the country can talk to their doctor. They can order it from Mass General. Across 8 different cardiovascular conditions, they've now got these risk scores. Of course, as well as being a collaborator, I'm also a patient of Pradeep's, because I go to the Preventative Cardiology Clinic because I'm someone who's obsessed with cardiovascular risk, right? So I got this test done. Turns out I don't have polygenic risk, so that's great. Haven't found any monogenic risk. The All of Us program didn't find any risk in my genome. So It's a lot of family history, and all I can do is essentially have a healthier lifestyle than I would say my father had in his 40s, where he was a smoker and he was overweight. I can avoid to as great extent I can those things, and then hopefully it looks like now my, risk of having a heart attack is lower than it could have been had I not taken those interventions, those lifestyle interventions. Other programs in the VA, so the Veterans Administration actually has another very interesting biobank known as the Million Veterans Project. And so they've been enrolling veterans into a biobank because veterans also have a set of healthcare issues, some of which are unique to them, to their population. And so with Jason Vassie, who's an investigator at the VA, we've launched a clinical trial in prostate cancer risk in the VA population, using the same sort of methods that we're using in the other programs. Also working with companies. There are companies who work directly with consumers now. Myome is an example. They've also been working with a research institute in Alabama to provide free genetic testing for common healthcare risks in the population in Alabama, because Alabama has one of the poorest healthcare outcomes in the country. So bringing this kind of cutting edge testing to the state of Alabama. And then other companies are offering this as well, using the same underlying technologies and algorithms and models that we've been building here. Okay, so I hope I've convinced you that there is a potential future in which the technologies that you heard about tonight could actually make a material impact on all of our healthcare. And I really would like to see us be able to get to a place where we spend, number one, we're not afraid to interact with the healthcare system. That the healthcare system is in fact interactive. That the insights, I haven't talked about AI and machine learning yet, ML, right? But almost everything I've talked about already incorporates some amount of AI and ML in it. The software that we use to look at the variants uses AI. The chips on the computers that we use to do the 4-hour genome or NVIDIA accelerated, machine learning chips. It's already part of what we're doing today, but there's more we can do with AI and ML. There's a whole bunch of hype and nonsense in AI and ML, but actually, fundamentally, it's just a really powerful tool that will help us sort through all of this information that we're gathering, that humans are just not equipped to manually sort through. When you think about a biobank, if you've got hundreds of thousands of people with hundreds of thousands of genomes, each genome has millions of variants, and you've got all these other pieces of information about their health and their lifestyle. A human can never make those associations and those patterns. It's exactly what AI is built to do. So AI is already making a difference. It's going to continue making a difference. The more we have linked data sets, larger biobanks, we'll be able to train better models and get these risks out there much more powerfully, much more predictive. And I think there is a possible world, and there's companies working on it, where you can have your genome sequence and you will be able to interact with that risk model and understand it. And I think it would be great if we spend as much time tinkering with and making small changes in our healthcare and our healthy aging plan as we do in our 401k. The Fidelity app is really easy to use, for the most part. If we had a little health app that that your hospital system or your insurance maintained, maybe not your insurance, but that somebody maintained that you had control over, that you could start to say, well, look at this risk over here. I should talk to my healthcare provider about additional screenings for these diseases. Because one of the things about saving money in the system is, if we all just go out and get screened for everything more often, that actually increases the healthcare cost burden. But by actually modeling your risk and say, well, actually, I'm not at higher risk for any one of these diseases than anybody else, but these two things I'm at much higher risk for, and I'm only going to really focus on additional screening for these things, we would end up saving the system money, okay? So the idea of interactivity, I think that would be a really great place to get. The healthcare systems themselves can also use this through their biobanks, there's fascinating stuff happening just looking at EHRs, the electronic health records, little signals in your labs, the notes that you have in your visits. Jordan Smoller, who runs some of the psychiatric program at Mass General, has published papers showing risk of bipolar disorder, things like suicidal tendencies, you can actually find signals of those things in people's medical records when you apply AI. So there's a tremendous amount that's going to happen in that. So that's sort of risk modeling and planning. Inevitably, when people have diseases, the other types of studies and technologies are going to help us have better therapies, more targeted therapies, not just in cancer, but in rare diseases as well. The ability to edit genomes is a tantalizing idea. And the idea that we can sort of, maybe hopefully bucket diseases together so that there are incentives to create therapies for more of the rare diseases. So to summarize, I guess my message was public investment in really difficult, audacious projects like the Human Genome Project have really returned a massive amount of value to us, both as a sort of a research community, but increasingly in the biomedical field and now getting into the healthcare system. I think that we're only going to see more improvements, more control, more ability to direct our own healthcare and our healthy aging with modern methods. And I think this is, and realizing that we can't just make this sort of the sport of kings for the people who can afford it, that actually the way to reduce the healthcare burden is to catalog this variation across all ancestries, all people, and make the sort of the risk mitigation available to all. That's where we're actually going to save money. And really it all comes from our continuing to believe and invest in science. Thank you.
Thank you so much, Niall. If you have any questions in the audience, raise your hand, and one of our mic runners will come over to you. But first, I'll just start off with, we have the science. We have the technology. We can make them better. What is it going to take to make sequencing from is it a regulatory thing? Is it a financial thing where we could just go to our doctor's office? Like if I have an appointment tomorrow, just say, hey, I'd like to get sequenced. I want to know what my risk is. I'm not a participant in a biobank. I actually am. But, how can that just become sort of the standard part of the care that we all get whenever we see our PCP?
Yeah, so I didn't get into it because it's a downer, and also it would take too long, and I'm not an expert. We have this issue of insurance, certainly in this country. And today, increasingly, insurance is covering genome sequencing, certainly in childhood rare diseases, in the NICU, in cancers where there's clear sort of actions and utility, you don't have issues. Convincing insurance companies that you could sequence the genome on a healthy individual and have that information about their risks and do prevention, and that would ultimately save them money, is a really tricky prospect the way that insurance works in this country, because a company is not necessarily incentivized to kind of help you avoid a disease 10 years from now, because most insurance companies have an individual customer for like 3 years. So they don't see they pay for the test, they don't see the benefit of the test, is their thinking. So we got to change that thinking to say that actually preventing disease helps them all, whether it's they pay for it, somebody else pays for it, they get the benefit of the test that somebody else paid for. That's the morass that has to be addressed. And I think the other thing that I did not get into, because it's a whole topic to be solved on its own, is of course sort of genetic privacy. Do you want your insurance company knowing your risks of disease? What would they do with that information? So we have to have the ability, I think, again, for individuals to control their own information, what's done with it. So there's a lot to address there.
Question over here. Oh, sorry. Go right ahead, sir. Sorry.
Yeah, I was really impressed by everything you said about how we're applying genetic variations, especially to newborn infants in the clinic. But 3 areas you didn't mention is one, monitoring somatic changes in diseases like cancer. 2, microbiome and tracing how that is related to health and disease and how that might vary even within an individual. And then RNA expression levels, transcriptomics in disease, and with testing back in less than a day. How close are we to being able to use that kind of information to trace the projects the progress of diseases and give us feedback that would help in the clinic?
Yeah, great question. So all of those things are happening. So somatic changes in cancer are just the variants in the tumors that I described. So I just didn't call them somatic variants. But that certainly is part of this sequencing of tumors and liquid biopsy. Microbiome is a fascinating, absolutely fascinating area, and I was involved in the Human Microbiome Project as well. And so there are lots of studies. Actually, in one of, we routinely at our lab are profiling gut microbiomes. And that is one of the big research areas that's happening at the Broad as well. We don't have a clinical test for microbiomes today, but there are companies locally, there are startups who are looking at microbiome therapeutics for things like C. diff and IBD. And so that's certainly an area. It uses exactly the same technologies I've described. And RNA, yes, so RNA, so it is in this sort of field of, again, multiple omics. And so we do a tremendous amount of RNA transcriptomics sequencing as well. In the All of Us cohort, we've just actually completed a pilot of 10,000 participants within the All of Us cohort, adding to their existing genome sequencing, transcriptome, RNA sequencing, and proteome, like protein profiling as well, to build up that picture of what is the value of adding these other omics to this for research, but also RNA sequencing is now often used in some of the rare disease diagnostics, because one way that you can understand if you find, say, 2 variants that might be causing the disease, and say that the way the disease shows up is a muscle disease or muscle wasting, one thing you can look at is in the RNA knowledge bases that we have, and these are other large public initiatives we've been involved in. GTEx is the Genotype Tissue Expression Project, so looking at RNA levels in different tissues. You could look at that patient and say, I found 2 variants in the genome. Let's look in the RNA level and say, well, of these 2 variants, which one of them is highly expressed in muscle? Because if that's the one that has the variant and the disease shows up as a muscle wasting disease, that helps us kind of get to those causative variants. So yes, all of those things are certainly happening. In our hands, at least, the microbiome and the RNA are not yet clinical tests, but there are clinical tests that use RNA and microbiomes in other groups.
Thank you for this amazing talk and summarizing the last 20 years of genomics in 1 hour. I'm also an ex-Broadie, and I started 20 years ago at the Broad. That was my first job. So big fan of the Broad Institute, obviously. My question is, if those programs become possible where everybody's sequenced in all the states, do we have the infrastructure for it? Where will the samples go? Where will they get processed?
Right.
How will the reporting be managed? What recommendation do you have if somebody were to ask you how to do that?
Yep.
You got
So
another
that's
hour?
Yeah, well, so that is exactly the kind of thing. So if you think about the newborn sequencing program, that's that Beacons program. That is exactly part of this implementation science, right? It's not, we technically know how to do it, but it's all of those logistics, right? It's the where did the sample get processed? How do we store the sample? It turns out that actually one of the big costs now, because the cost of sequencing genomes has become so low, storing the data, when you sequence a genome, you create a lot of data, like more than you can keep on a laptop. And so actually storing all of that data becomes quite expensive. So there's data management becomes a legitimate issue. And also just, again, we have to be so careful about data security, right? And one of the actually interesting things about the way the All of Us Biobank program was set up is that, sort of in a prescient way, was that when it was set up many years ago, participants who have their genome sequenced as part of All of Us, the data is stored in a cloud environment, in a secure environment, but it's actually, even though the project is funded by the NIH, the government does not own the data. The government doesn't have access to the data. The data is stored by an awardee, actually it turns out to be Vanderbilt University, is the curator of the data. So they're responsible for keeping it safe and access to it. And actually, they have what's called a certificate of confidentiality, which means the government cannot access that data for law enforcement purposes, for other things like that. So those kinds of things those walls are very important to create to build confidence, privacy, security. So all of that logistical things do have to be worked out. That is what programs like Beacons and others are trying to do. And there's lots of companies who are also trying to do this. And it will be interesting to see, of course, there's a lot of public effort. There's also a bunch of private effort. And so there's a lot of models out there where you could go to a company and get your genome sequenced. And one model is, and then likely, again, this is for people who can pay for it. There is sort of the subscription model. You could have your genome sequenced and have it accessible whenever you need to access it, or something happens and you want to have someone interrogate it, you could do that on demand, right? So different models are showing up, whether in the public system, the healthcare system, or in sort of private companies.
And actually, that segues nicely into a question that we actually had somebody ask on YouTube was, you mentioned that a researcher comes to the Broad and they can get their sample sequenced for around $350. I imagine that depends on volume or scale, but if your average person off the street wanted to get their genome sequenced, what would that run though?
Yeah, so again, so we don't sort of do this sort of, this is kind of like the direct to consumer model, right? Of just sort of people coming and paying for their genome to be sequenced. That's not what we do. Genomes that we sequence are usually part, again, of these larger research studies that we describe. In the clinical world, where we do clinical whole genome sequencing, it's like any other clinical test. It comes from an order from a healthcare provider. So individuals can't sort of just send us their sample and ask us to sequence their genome, but there are companies who will do that, right? So there's lots now of direct-to-consumer companies who offer, the same kind of health risks, whether it's monogenic, polygenic reports, back to consumers. And you can actually just go and have a company do it, and they're typically running about $500 to start. Different models, but yeah.
Question here, and then we had somebody up in the middle as well. So start with you.
Thank you. Hi, Dr. Niall. I'm Yan Yan. I'm currently a student from HMS, and I'm an Aspire founder. So I have a two-part question. First question is on the, I want you to kind of walk us through like a patient process for somebody who wants to get, for example, the cardiovascular genomic risk assessment. I know we had a breakthrough now of very fast, under 4-hour genome sequencing, but I can't help but to think the genome interpretation part of it is still catching up. So could that be a potential bottleneck in the future of having like readily write out of your genomic assessment risks? So that's my first part of the question. Second is on the genetic privacy that you mentioned. In the future, could there be possibly this centralized model where a patient will have control of their own genetic information and they have the power to monetize it themselves rather than having some company start up monetizing on their data in fine print, right, when you sign a paper with them? So that's my question. Thank you.
Yep. So on the first part, I think, if I understand the question, it was sort of the, even if we can sequence the genome faster, assessing the genomic risk is going to be slower, and that's a bottleneck. That's going to be true. And I would say, for the most part, unless you're in the NICU and some other settings, we don't necessarily need to sequence your genome in 4 hours, right? If you're going to do a genomic risk assessment, your risk, we're talking about years of like lifetime risk, right? And so you're really, if that report takes 3 weeks or 6 weeks to get back to you, it's unlikely, a major chronic disease will have sort of magically shown up in that time frame. So I think matching the technology capability with the sort of the clinical need, will happen naturally. And I think some things don't need to be massively fast, some things do. I think on the second part, so the question being, do you think there's a future where people will be able to have their genome sequenced and sort of control it or monetize it themselves? It is an interesting idea, and lots of folks have been thinking about ways to enable that. I think, again, those are going to be companies. Somebody has to actually create the entity that allows you to do that, right? It's not clear whether a healthcare system is going to be set up to do it. Insurance companies are not necessarily going to be the ones to do it. So I think it will end up being some version of sort of, public-private startup or something else that would maybe create a model that allows you to have your genome sequenced. You might have to pay them some nominal fee, but then I didn't really touch on it, but one of the very interesting things about calculating lifetime risk of disease, one of the other use cases could be what's known as patient stratification, which essentially, if you want to do a clinical trial today as a pharmaceutical company on a drug that might help you avoid getting a cardiovascular disease, to get the statistical power to decide has your drug done something, you need to enroll like tens of thousands of people. And then you've got to wait for a really long time and say, well, how many people got a heart attack and how many people didn't? It takes a really long time, and they're massively expensive clinical trials, and then it might not work. So this is like the cost of developing drugs. If you were to take a whole bunch of people, run a really powerful risk model, and say, well, actually, these are all the people who are at like 5x risk of having a heart attack in the next 10 years. Let's enroll them. Then we actually have to we can stratify, the trial and have a much smaller trial and have it be more powerful and actually be able to see if there's an effect sooner. So that's the kind of idea of the promise of also being able to model risk. And so there could be models where in biobanks, through this information, that people could get identified as being important to enroll or try to enroll in a clinical trial. And then there are companies who've talked about this idea of like, well, if you do get, pharmaceutical companies will pay to find people who can be in their trial. So there could be this idea that people could sort of, through a tokenized way, have their data available, and if somebody wants to enroll them in a trial, that they have the ability to sort of benefit from that as well. I would say it's not a mature system yet, though, but people certainly have been considering that.
I think we just found a new use for the blockchain. Last question from the gentleman in the middle.
Right. I am not a Brody, and I'm not living in the genomic world based on my own scientific education, but it appears that we're in a political environment where public support of science is changing, or perhaps changing. Do you, in your opinion, see private or a commercial groundswell or investment that might grow or to equal or overtake that, so that all these things you're talking about that individuals might hope for to increase their own and be involved in a more fulsome level in their own medicine, do you see there a possibility of individuals and the people being able to take over and fund this, so we're not reliant on a changing public support system?
Yeah, it's a really important question, and there's no easy answer. I think so a couple of things have happened. I would say I think you're right, and I think certainly us, those of us who've been working in this area, we didn't really mention it here, but during the pandemic, the Broad's Clinical Lab did actually do a lot of the COVID testing that happened in New England. And that was happening in our clinical lab. We did 37 million tests. And so we felt like this great energy that we were able to contribute. I think what we've seen, or at least what I've observed post-pandemic, was there has been a little bit of sort of separation in the population of people who believe that how we as a country handled the pandemic was completely wrong, and people who believe we did what we could do. And that has sort of, I think, undermined a little bit of the belief in science in some sections of the population. I think, unfortunately, it also sort of conflates science with public health policy, which is a totally different field. I would not want to be in setting public health policy for anybody. But so I think that's right. I think those of us who work in science do worry that has been a reality. I think, and as the current climate has rolled out, we have certainly seen funding challenges in areas like NIH and CDC and others, and that's been a concern. There has been groups who have stepped up, and certainly, as I mentioned earlier, a lot of the projects we do, they have federal funding, they have philanthropic funding, pharma, and so those other sources of funding have certainly continued to exist, and in some cases have increased. However, I use the shuttle launch and the human genome as examples. I think it's very unlikely that we'll find another source as powerful as the federal government. The federal government is the aggregate of all of the funds that we sort of pay through our taxes. They have the deepest possible pockets. No one company is going to step up and fill that gap. And so we do need to continue to advocate that we as a country sort of invest in science. And and we at the Broad have now a really talented sort of government relations team. And I think what we're finding is that you hear a lot of stuff in the news, but people in red states and people in blue states get sick. They have sick children, they have rare diseases, they get cancer. Healthcare, everyone feels the pain. And everyone would prefer not to get sick, to have a better treatment for their family if they get sick. So there is, by and large, I think, bipartisan support for continuing to invest as a country in science. It may not show up on the news, but I think what we're hearing is that there is support for that. And that people believe that things like biobanks are important and that there might be support across the aisles in that. So I think I'm not I'm certainly not a political expert, and I try to avoid the news as much as possible. But I've got to believe that the stuff that we talked about tonight is important to everybody, regardless of their political affiliation. And we're going to continue to look at ways to, bridge the gaps that we think hopefully will be short term in any funding with these other sources to get us to the point where we can keep proving the benefit of what's happening and that it actually is in all of our benefits to see these things through.
And I think that is the perfect note on which to end the evening. Thank you to all of you for joining us tonight. Thank you to everybody watching online, and thank you, Niall.